ಪ್ರಿಯ ವಿದ್ಯಾರ್ಥಿಗಳೇ, ಜಿಲ್ಲಾ ಪಂಚಾಯತ್ ಉಡುಪಿ ಇವರು ಆಯೋಜಿಸಿರುವ CET ಹಾಗೂ ನೀಟ್ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಈ ದಿನದ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ನಾವು we are going to discuss the biotechnology and its principles, procedures. ಹಾಗೆ ನೆಕ್ಸ್ಟ್ ಸೆಷನಲ್ಲಿ ಬಯೋಟೆಕ್ನಾಲಜಿ ಆ್ಯಂಡ್ ಇಟ್ಸ್ ಅಪ್ಲಿಕೇಶನ್ಸ್ ಈ ಬಗ್ಗೆ ನಾವು ಸ್ವಲ್ಪ ವಿಶದವಾಗಿ ಕಲಿಯೋಣ. Now this is a concept based chapter. Biotechnology, if you know the concept very well, in the competitive examination you can do well. So you have to know the concept. Okay, it is a concept based chapter. Once you know the concept, it is very easy to solve any type of questions. It is not a memory based concept. Okay, now let us try to understand a brief account of what is the biotechnology principles and the processes. You know, the definition for biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. That is the definition that is there in your NCERT book. So I can define in another way. It is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. Application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. So as far as me is concerned, this is the best definition that related to the biotechnology. Now, when we talk about biotechnology, it began when man invented the art of doing, that is, beverages, bread, like that. So that we call it as traditional biotechnology. In traditional biotechnology, whole organisms are used. There is no modification of organisms. But the two core areas, core techniques of modern biotechnology are that gave birth to modern biotechnology also. One is genetic engineering. Okay, genetic engineering technique to alter the chemistry of genetic material, like DNA and RNA. Second one is the bioprocess engineering, maintenance of sterile ambience in chemical engineering to process to enable only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, like that. So these two core areas gave birth to modern biotechnology, you know that. So these two core areas, one is the technique to alter the chemistry of DNA and RNA, and then maintenance of sterile ambience chemical environment. That means only desired organisms can be grown and undesired organisms can be eliminated. Now, when we talk about biotechnology, broadly we can divide the two concepts. One is, just imagine a patient is there in the hospital. To operate, we require certain tools and then certain procedures we have to follow for operations. So let us study what are the tools that are required for rDNA technology, because the main area of biotechnology we are concerning with creating rDNA, recombinant DNA technology. It is also called genetic engineering. It is also called gene cloning. So for gene cloning, genetic engineering, or to create rDNA, that is recombinant DNA technology, we require certain tools. The tools are, see, enzymes. Among the enzymes, restriction endonuclease is the most important tool that is required. Restriction endonuclease. Then enzyme ligase, ligase is used to join the any two cut ends of DNA, and then DNA polymerase for cloning the desired gene that is required. So restriction endonuclease, ligase enzyme, DNA ligase, and then DNA polymerase are the three enzymes. Besides these three enzymes, there are other enzymes also, but these three enzymes are very, very important. Then vectors are required. Vectors are the DNA molecules that carry a desired gene from choice organisms to organism of interest. Vectors. So the most commonly used vector in creating rDNA or rDNA technology, plasmid or bacteriophage. Even cosmids can also be used. So plasmids and bacteriophages are most commonly used vectors in rDNA technology. Then for cloning the desired gene, we require host. That host, most commonly used host is Escherichia coli, a bacteria. Okay. Even plants and animals could also be used as a host. Then, bioreactors. Bioreactors are large vessels, fermenters, where desired products can be produced in large quantities. We will see one by one when it comes to the MCQ. I serially arranged all the MCQs. Along with the MCQ, we will discuss. Okay. Then, processes, the broad outline procedures that we follow during the biotechnological operations. They are isolation of DNA by various enzymes. For example, if we use the DNA from a plant cell, we have to use cellulase enzymes to degrade the cell wall. If we use the DNA from fungi, we have to use chitinase, okay, fungal cellulase, that is the enzymes to degrade the fungal cellulose. If we use bacteria from which DNA is required, suppose we have to use lysozyme, that is the enzyme that degrade the bacterial cell wall. Then after degrading the cell wall, then we have to treat the cell with various enzymes like proteases, lipases, okay, amylases, RNases. All the macromolecules that are present in the cell must be degraded. ಸೆಲ್ನಲ್ಲಿ ಇರುವಂಥ ಎಲ್ಲ ಮಾಲಿಕ್ಯೂಲ್ಸು, ಪ್ರೋಟೀನ್ಸು, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸು, ಹಾಗೆ ಲಿಪಿಡ್ಸು, RNA ಇವುಗಳನ್ನೆಲ್ಲ ನಾವು ಮೊದಲು ತೆಗಿಬೇಕು. ಅದನ್ನು ತೆಗಿಬೇಕಾದ್ರೆ ಅಪ್ರೋಪ್ರಿಯೇಟ್ ಎಂಜೈಮ್ಸನ್ನು ನಾವು ಯೂಸ್ ಮಾಡಬೇಕು. ನಾವು ಆಫ್ಟರ್ ಟ್ರೀಟಿಂಗ್ ಆಲ್ ದಿಸ್, ಯು ಡೋಂಟ್ ಯೂಸ್ DNase ಎಂಜೈಮ್. ದಟ್ ಈಸ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. DNase ಎಂಜೈಮನ್ನು ಯೂಸ್ ಮಾಡಿದ್ರೆ DNA ಡಿಗ್ರೇಡ್ ಆಗಿ ಹೋಗ್ಬಿಡುತ್ತೆ. ಸೊ ಎಕ್ಸೆಪ್ಟ್ DNase, ಆಲ್ ಎಂಜೈಮ್ಸ್ that are appropriate to treat the cell have to be used. Okay, that is the isolation of DNA. Then pure DNA comes in a chilled ethanol. Okay, that is precipitated in chilled ethanol. The next step is fragmentation of DNA by restriction endonuclease. Restriction endonuclease, in a rDNA technology we call it as molecular scissors. So these cut the DNA within the DNA segment by disturbing the phosphodiester bonds to make a fragments. You know that when it comes to the endonuclease, there are class of enzymes called nucleases. So nuclease acts on nucleic acids. There are two class of nucleases, exonucleases and endonucleases. Exonucleases are the enzymes that removes the nucleotide from terminal position. They are not used in biotechnology. Whereas the endonuclease enzymes, they cut internally in DNA, cutting the phosphodiester bonds. One particular class of enzymes that we use in biotechnology, we call it as molecular scissors, are restriction endonuclease. Now, why they are called restriction endonuclease? Because these endonucleases, restriction endonuclease, were present in bacteria. They restrict the growth of viral multiplication. They restrict the growth of virus by degrading the DNA of virus, bacteriophages. That's why it is restriction endonuclease. Now, the first restriction endonuclease was isolated was Hind second. Okay, Hind second. Later, several restriction endonucleases were isolated. Now already we have more than 900 such restriction endonucleases obtained from over 230 different strains of bacteria, and each restriction endonuclease has named, and that naming is very very important from the point of view of competitive examination that we will study when it comes to MCQ ಬಂದಾಗ ಅದಕ್ಕೆ ಸ್ಪೆಸಿಫಿಕ್ ಆಗಿ ನಾವು ಸ್ಟಡಿ ಮಾಡೋಣ. ಸೊ ಟೋಟಲಿ ದಿ ಸೆಕೆಂಡ್ ಇಂಪಾರ್ಟೆಂಟ್ process that is involved in biotechnology is fragmentation of DNA by restriction endonuclease. The third step is the isolation of desired DNA fragment. From the various fragments are generated by restriction endonuclease digestion, we have to separate and isolate the DNA fragment. Separation of desired DNA fragment from the gel electrophoresis. So gel electrophoresis used to separate the DNA fragments, that we will study when it comes to MCQ. Then selection of suitable plasmid and cleaving by using restriction endonuclease. So suitable plasmid, that is molecular vehicles, should be used to carry the desired gene from one organisms to another organisms. Next, ligation of the DNA fragment into a vector. This is the most important aspect. We call it as gene splicing. ನಮಗೆ ಡಿಸೈರ್ಡ್ ಡಿ ಎನ್ ಎ ನಾವು ತೆಕ್ಕೊಂಡು ಅದನ್ನು ಕ್ಲೀವ್ಡ್ ಪ್ಲಾಸ್ಮಿಡ್ಗೆ ಸೇರಿದಾಗ ಅಲ್ಲಿ ರೀಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ದಿ ಸೆಂಟ್ರಲ್ ಆಸ್ಪೆಕ್ಟ್ ಆಫ್ ಕ್ರಿಯೇಟಿಂಗ್ ದಿ ಆರ್ ಡಿ ಎನ್ ಎ ಟೆಕ್ನಾಲಜಿ, ದಟ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಜೀನ್ ಸ್ಪ್ಲೈಸಿಂಗ್, ಜಾಯ್ನಿಂಗ್ ದಿ desired DNA segment with the cleaved plasmid to form rDNA. This is achieved by using DNA ligase enzyme. You know, DNA ligase enzyme, we call it as molecular glues or molecular stitchers. Okay. The next step is transferring the recombinant DNA into host cell. You know, this procedure we simply call it as transformation. So during transformation, the rDNA enter into the host cell. Okay, only 10% or below 10% of the rDNA enter into the host cell. Next very very important procedure that we have to follow is selection of transformants. Selection of transformants, because not all the host cells will take the rDNA, only some will take the rDNA. These host cells that take the rDNA are called transformants. Those host cells which do not take the rDNA are called non-transformants. If you grow this non-transformant in bioreactor, it is a wasteful process. So we have to grow only transformants. So for that we have a selection of transformants. There are two methods we do for selection of transformants. Is one by using the antibiotic resistant sites or by insertional inactivation. So either one of the methods we select the transformants. Then after that, culturing the host cells in a medium on a large scale. Okay. Then finally the extraction of the desired product. So these are briefly the procedures, operational procedure that we have to follow during the creation of rDNA technology, rDNA. So that is the main procedures involved in recombinant DNA technology. Okay. Now, with this brief introduction, we will move on to some questions, MCQ questions, okay, and try to answer what are the relevant answer for these questions. Okay. See, which one among the technique is involved in modern biotechnology? The question is, which one among the technique is involved in modern biotechnology? Option A, making curd from milk by lactobacillus. Option B, making bread from dough by yeast. Option C, making insulin from Escherichia coli through gene cloning. And option D, making wine from fruit juices by yeast. You know, the A, B and D are the traditional biotechnology, where the organisms that we use is unaltered. In modern biotechnology, we use organisms that were altered. Altered organisms are used, manipulated organisms are used. Organisms are manipulated by cutting, joining and cloning these organisms to produce desired products in large quantities. That is the main thrust of the modern biotechnology. Okay, therefore the option is C. Option is C, making insulin from Escherichia coli through gene cloning. So this is the right answer. Okay, so the right answer for which one among the technique is involved in modern biotechnology is making insulin from Escherichia coli through gene cloning. Gene cloning. Okay. See, this is the brief account of procedure that I have depicted in the form of a labeled flow chart for the production of human insulin. Okay. So here restriction and enzyme is used to cut the desired DNA from choice organisms, and DNA ligase is used to join the desired gene with the cleaved plasmid to form recombinant DNA. This is the recombinant DNA, and then this gene is cloned. So the procedure involved for insulin production is quite different, that we will study when it comes to the next chapter, that is biotechnology and its applications. Okay. Then move on to the next question. See, select the incorrect statement with respect to rDNA technology from the following option. So we have to select, ಇಲ್ಲಿ ಒಂದು incorrect statement ಇದೆ, 3 are correct statement ಇದೆ, ಒಂದು incorrect statement ಇದೆ. ಯಾವುದು incorrect statement ಅಂತ ನೋಡೋಣ. The first instance of creating the rDNA was done by Stanley Cohen and Herbert Boyer in 1972. Okay, so that is exactly right. It is right. This is right. So there is no mistake in the sentence. ಈ sentence ಅಲ್ಲಿ ಯಾವುದೇ mistake ಇಲ್ಲ. First the rDNA was created. rDNA was first created by Stanley Cohen and Herbert Boyer in 1972. That is correct. Next, the first restriction endonuclease isolated was Hind second. Hind means Haemophilus influenza. Strain number D, and that is the Roman letter 2 means the order of isolation that we will come across when it comes to appropriate questions. So that's also a correct. Okay, the first restriction endonuclease isolated was Hind second in 1963. Arbor and Smith isolated this, and that gave the modern biotechnology, that will push the modern biotechnology. Next, third, the recognition sequence for Hind second has 8 base pairs. So look at this option. The recognition sequence for Hind second has 8 base pairs. What is recognition sequence? Recognition sequence andre DNA alliruva particular sequence. Okay, we call it as palindromic sequences. Sequences having reverse repeats, two-fold symmetry. If you read both backward and forward, it has the same nitrogen base sequence. Such sequences that are present in the DNA, we call it as recognition sequences. The recognition sequences for Hind second, is GAATTC, GAATTC. On the 5' to 3' direction, it has GAATTC. If you read from backward to forward, from 5' to 3', it has GAATTC. So therefore, the recognition sequence for Hind second has not 8 base pairs, it is 6 base pairs. So that is the answer. So therefore, the incorrect statement is C. Okay, Hind second was isolated from Escherichia coli. That's also here it is wrong. Okay, so here 2 statements are wrong there, just it is a mistake by me. One is the recognition sequence for Hind second has 6 base pairs, and Hind second was isolated from the Haemophilus influenza. So it is not the Escherichia coli. Okay, so both statements are wrong here. Okay, you make note on. Okay, so this is the recognition sequence. Okay, then we move on to next question. Match the following. We have E, CO, R and I in column 1, and in column 2 we have indicates species, indicates genus, indicates order of isolation, and it indicates strains. So what this, that each restriction endonuclease that isolated from the bacteria is named according to some convention. The first letter of the restriction endonuclease is taken from genus from which it is isolated. Next two letters is taken from species from which it is isolated. Okay. The next strain number that we use, example R, D, S, like that. Then Roman letter indicates order of isolation. So on that basis, so see E. E means it indicates genus. So AQ. AQ. Okay. CO. That means indicate species. Next two letters are used to indicate species. Then R indicates the strain. Okay, R indicates strain. Then the Roman letter first here indicates the order of isolation. So therefore the option you see here AQ, BP, CS and DR. So this is the option. Option B is the correct answer. Okay. So see here, E genus, CO species, R is the strain and Roman letter order of isolation. Okay. Then we move on to next question. Okay. Select the incorrect statement with respect to action of restriction enzymes from the following option. Option A, see here, exonucleases removes nucleotides from the ends. Okay. Option B, endonucleases makes cuts at specific position within the DNA. Option C, each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. And D, endonucleases cut the two strands of the DNA at specific points in their hydrogen bonds. So these are the four options. We have to select the incorrect options. ಈ ನಾಲ್ಕು ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಲ್ಲಿ ಎಲ್ಲಿ ಇನ್ಕರೆಕ್ಟ್ ಇದೆ ಅಂತ ನೋಡಬೇಕು. See, exonuclease removes nucleotides from the ends. That is correct. Nucleotides are removed. You already know certain enzymes, for example, exopeptidases with respect to proteases enzymes. They remove amino acids from the terminal position. Okay. Endopeptidases on the other hand, they make internal cuts in the polypeptide chains. Similarly, we have exonucleases. These are the enzymes that remove the nucleotides from the ends. Whereas endonucleases make internal cuts. Endonucleases makes cuts at a specific position within the DNA. So both statements are right. First, A and B are right. Next, each restriction endonuclease recognizes specific palindromic nucleotide sequences in the DNA. That is also exactly right. So first three statements are right. Then we come to D. Endonucleases cut these two strands of DNA at specific points in their sugar phosphate backbone. Okay, sugar phosphate backbone anna cut ಮಾಡುತ್ತೆ, not in the axis. You know, DNA is made up of axis that constitute hydrogen bonds. So between adenine and thymine, 2 hydrogen bonds are there. Between guanine and cytosine, 3 hydrogen bonds are there that makes up the axis of the DNA. But the backbone of each polynucleotide strand made up of repeating units of sugar and phosphate, that we call it as phosphodiester bonds. That phosphodiester bonds are broken by these endonucleases. So therefore the incorrect option is fourth. So what is the correct statement that we make from the D? Endonucleases cut the two strands of DNA at specific points in their phosphodiester backbone, phosphodiester bands. Okay. Then we move on to the next question. So this is the how the endonuclease cuts the DNA internally, leaving sticky ends. So they cut the DNA at sugar phosphate backbones, phosphodiester bonds, and making the sticky ends. See here, this the sticky ends. And these sticky ends have complementary base pairs. You know complementary base pairs? Adenine always pairs with thiamine with 2 hydrogen bonds, and guanine always pairs with cytosine with 3 hydrogen bonds. This type of base pairing is called as complementary base pairing. So the restriction endonuclease cuts the DNA, leaving behind sticky ends. So these sticky ends could be joined by using DNA ligase. Okay. Next question. See, find the recognition sequence for Escherichia coli restriction 1 endonuclease. Recognition sequence. You know, you have to remember from the competitive examination point of view, ಎರಡು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಅನ್ನು ನೀವು ತಿಳ್ಕೋಬೇಕು. ಎರಡು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ತಿಳ್ಕೊಂಡ್ರೆ ಸಾಕು. ಹಾಗೆ ಒಂದು ವೆಕ್ಟರ್ನ ತಿಳ್ಕೋಬೇಕು. ಹಾಗೆ ಒಂದು ದಿ ರಿಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಅನ್ನು ನೆನಪಿಟ್ಕೊಂಡಿರಬೇಕು. ಸೊ ಒಂದು ಎಂಜೈಮ್, ಯು ನೋ, ಇಟ್ ಈಸ್ ಇ ಕೋಲೈ R1 restriction endonuclease that is obtained from Escherichia coli. Second one, you know, the first restriction endonuclease isolated was Hind second. And also you have to know one more restriction endonuclease, that is BOM H1, very very important. Bacillus amyloliquefaciens H BOM H1, order of isolation first. So these three restriction endonuclease you have to remember from the competitive examination point of view. Two vectors you have to know. One vector, you know, PBR322, another one is PUC18. So these are the two commonly used vectors. So but preferably the PBR322 you have to remember. Okay. We will come across related to that question also. See, in this question, find the recognition sequence for Escherichia coli restriction 1. See, the option 1 is 5' to 3'. We have nucleotide sequences as GAATTC. Okay. So if you read the nucleotide sequence from the other strand from 5' to 3' direction only, that means orientation of the reading should be kept same. That is very very important. We have the same nucleotide sequences GAATTC. So both forward as well as backward, it has same nucleotide sequence. So that is the 6 base pairs, 1, 2, 3, 4, 5, 6 base pairs. GAATTC, 6 base pair is the recognition sequence for Escherichia coli. ಅಂದರೆ ಅದರ ಅರ್ಥ DNA ಸ್ಟ್ರ್ಯಾಂಡಲ್ಲಿ, ಲಾಂಗ್ DNA ಸ್ಟ್ರ್ಯಾಂಡಲ್ಲಿ GAATTC ಈ ಸೀಕ್ವೆನ್ಸ್ ಇದ್ದರೆ ಏನು ಮಾಡುತ್ತೆ? ದಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ Escherichia coli restriction 1 endonuclease cuts, leaving behind sticky ends. Suppose, just imagine, if the DNA strand has 2 such regions, it makes 2 cuts. If it has 3, it makes 3 cuts. If it has many, how many recognition sequences are there, and that region it will cuts. Suppose if there are no recognition sequences, ಒಂದು DNA ಅಲ್ಲಿ ಆ recognition sequence ಇರೋದಿಲ್ಲ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ, it will not cut. Suppose if you use restriction endonuclease Escherichia coli restriction 1. So the option is this one. All others are not palindromic sequences. You see, the option B, option C and option D are not recognition sequences because they are not palindromes. Okay, they are not reverse repeats. Okay, so the option is first GAATTC. ikkadindaru ನೀವು read ಮಾಡಿದ್ರೆ it has same nucleotide sequence GAATTC. Okay, so this is the GAATTC is the recognition sequence for Escherichia coli restriction 1. So this is the recognition sequence for Hind third, Haemophilus influenza, strain number D and order of isolation third. See, it cuts here, leaving behind sticky ends. And for BOM H1, Bacillus genus, AM amyloliquefaciens, amylo species. H is the strain number and 1 is the first, that is the first it is isolated. So its recognition sequence are GGATCC. GGA-TCC. So it cuts here. So usually the restriction endonuclease cuts away, slightly away from the center, but on same point on both the strands. So leaving behind streaky ends. Okay. Then see the next question. Question number 6. Read the statement A and B. Here we have two statements. Okay, statement 1, statement 2. Choose the right one. Suppose if the both statements are right, ಎರಡೂ ಸ್ಟೇಟ್ಮೆಂಟು ಕರೆಕ್ಟ್ ಇದ್ದರೆ ನಾವು ಆಪ್ಷನ್ನು 2 ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕು. ಓಕೆ, ಆಪ್ಷನ್ 2 ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕು ಈ ಕೇಸಲ್ಲಿ. ಸಪೋಸ್ ಇಫ್ ದಿ ಸ್ಟೇಟ್ಮೆಂಟ್ ಬಿ is the right answer for the, that means reason for the statement 1, we have to choose option 1. ಓಕೆ, ಎರಡೂ ಸರಿಯಾಗಿದ್ದು ಎರಡನೇ ಸ್ಟೇಟ್ಮೆಂಟು ಫಸ್ಟ್ನೇ ಸ್ಟೇಟ್ಮೆಂಟ್ಗೆ ರೀಸನ್ ಆಗಿದ್ದರೆ ಫಸ್ಟ್ ಆಪ್ಷನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕು. ಎರಡೂ ಸ್ಟೇಟ್ಮೆಂಟು ಸರಿ ಇದ್ದು ಎರಡನೇ ಸ್ಟೇಟ್ಮೆಂಟು ಫಸ್ಟ್ನೇ ಸ್ಟೇಟ್ಮೆಂಟ್ಗೆ ರೀಸನ್ ಆಗಿಲ್ಲದೆ ಇದ್ದಾಗ ವಿ ಹ್ಯಾವ್ ಟು ಸೆಲೆಕ್ಟ್ ಆಪ್ಷನ್ ಬಿ. ದೆನ್ ಫಸ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ಸ್ ರೈಟ್, ಸೆಕೆಂಡ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಫಾಲ್ಸ್, ರಾಂಗ್ ಇದ್ದಾಗ ಥರ್ಡ್ ಆಪ್ಷನ್ನು, ಎರಡೂ ಸ್ಟೇಟ್ಮೆಂಟು ರಾಂಗ್ ಇದ್ದರೆ ಫೋರ್ತ್ ಆಪ್ಷನನ್ನು ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕು. ಓಕೆ, ಸಿ ದಿ ಸ್ಟೇಟ್ಮೆಂಟ್ 1, DNA ligase, you know DNA ligase is called molecular glues. Okay, in genetic engineering is called molecular glues. Okay, that is the statement. So this statement is right. Then we will move on to statement 2. DNA ligase is used to join the two DNA fragments with the sticky ends. So that is the statement 2. Here both statements are right and the statement 2 is the right answer for the statement 1. Okay, therefore we have to select the option A. Both statement A and B are correct and statement B is the correct explanation of the statement A. Okay, next we move on to another question, that is which statement is correct with regard to plasmids. Okay, now we concentrate some few points with regard to plasmids. You know, plasmids are autonomously replicating, self-replicating. Their replication is independent of the bacterial chromosome. Second, they are double-stranded, circular, naked DNA present in the cytoplasma of many bacteria. One thing. Now, all bacteria do not have these plasmids, but majority of bacteria, most of the bacteria do have plasmids. Okay, they are self-replicating or autonomously replicating, double-stranded, circular, naked. Naked means they are not associated with histone proteins. They are circular, not linear. Okay, circular, naked, double-stranded, autonomously replicating DNA present in the cytoplasma of many bacteria are known as plasmids. You know, plasmids are used as carrying desired gene from organism of choice into the organism of interest. Here organism of interest is host. Okay, from the choice organism we isolate the desired gene and this desired gene is incorporated into the cleaved plasmid and that we create the rDNA and rDNA is introduced into the host cells. So that is the procedure that we follow in the rDNA technology. And with regard to the plasmids, see what are the options. Self-replicating linear extra chromosomal DNA. It is extra chromosomal DNA, okay. It is self-replicating, okay, but it is not linear. It is not linear. It is not linear DNA. So linear DNA found only in eukaryotes. Okay, it is found in eukaryotes. Then B, self-replicating circular chromosomal DNA. It is not chromosomal DNA. You know, bacteria have main chromosomes, bacterial chromosomes. Chromosomal DNA, here also they are not linear, circular, they are lacking histone proteins, all you know that. So it is not chromosomal DNA, second option is also wrong. Then self-replicating linear chromosomal DNA, once again the word linear is there, so it is linear alla, it is circular. Then self-replicating circular extra chromosomal DNA is the correct answer for plasmids. Okay, option 4th is the correct. Then we move on to next question. So see here, The bacterial DNA, these are the plasmids. Okay, plasmids are present in the cytoplasm of many bacteria, and you know that their number vary from bacteria to bacterial species. One species in the ಇನ್ನೊಂದು species number, it depends upon the copy number that is associated with the ori that we will come. Okay, then see, diagrammatic representation of rDNA technology. Diagrammatic representation of rDNA technology is given from these. Select the correct option among the following. See, we have A, we have B here, we have C, and we have D. So these are the options. These are the alphabetical markings on the flow chart. What these alphabetical marks indicate? Let us study. So you know that A here is the restriction endonuclease that is used to cut the desired gene from the isolated DNA. Okay, so A is REN. A is REN, restriction endonuclease. B is also restriction endonuclease because it is used to cleave the plasmid. And one thing you have to remember, you know, the joining is made possible. ನಾವು ಡಿ ಎನ್ ಎ ಲೈಗೇಸನ್ನು ಯೂಸ್ ಮಾಡಿ ಜಾಯಿನ್ ಮಾಡ್ತೀವಲ್ಲ, ಪ್ರೊವೈಡೆಡ್ ಆ ಜಾಯ್ನಿಂಗ್ ಆಗಬೇಕಾದ್ರೆ ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಇರಬೇಕು. ಓಕೆ, ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಯಾವಾಗಿರುತ್ತೆ? ಇಫ್ ವಿ ಯೂಸ್ ದಿ ಸೇಮ್ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಟು ಕ್ಲೀವ್ ದಿ ಡಿಸೈರ್ಡ್ gene from choice organisms from DNA, and also same REN is used to cleave the plasmid, then only it creates this sticky ends. Sticky ends ಅನ್ನು ಕ್ರಿಯೇಟ್ ಮಾಡಬೇಕಿದ್ರೆ ಮತ್ತು ಜಾಯಿನ್ ಆಗಬೇಕಿದ್ರೆ ನಾವು ಸೇಮ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸನ್ನು ಯೂಸ್ ಮಾಡಬೇಕು ಟು ಕಟ್ ದಿ ಡಿ ಎನ್ ಎ ಫ್ರಮ್ isolated DNA from the long strands of DNA, and also we have to make cut in the plasmids to create sticky ends. So therefore A and B should be restriction endonuclease. Now C, you know, it is very simple. It is the DNA ligase that is used to join the desired gene with cleaved plasmid to form rDNA. What is rDNA? You know that it is the vector containing foreign gene, vector containing alien gene, vector containing desired gene. So that is called as rDNA. Then you know D. What is D here? This rDNA does not undergo multiplication if they are not introduced into the host cell, because this rDNA is incorporated along with the origin of replication of the plasmid. So in order to multiply, the rDNA must enter into the host cell. rDNA na host cell ge introduce ಮಾಡಬೇಕು. That phenomenon of introduction, incorporation of the rDNA into the host is known as transformation. So therefore the option here, both A and B are REN, and C is the DNA ligase, and D is the transformation. Okay, then we move on to next question. The criterion for DNA fragment movement on agarose gel during gel electrophoresis is, you know, gel electrophoresis is the procedure technique that is used to separate the DNA fragments. Okay, DNA fragments are generated out of restriction endonuclease digestion. ಇಲ್ಲಿ ಜನರೇಟ್ ಆದಂಥ ಫ್ರಾಗ್ಮೆಂಟ್ಸು ಕೆಲವು ಚಿಕ್ಕ ಇರುತ್ತೆ, ಕೆಲವು ಮೀಡಿಯಮ್ ಸೈಜ್ ಇರುತ್ತೆ, ಕೆಲವು ಲಾಂಗ್ ಇರುತ್ತೆ. ಸೊ ಬೇಸ್ಡ್ ಆನ್ ದಿ ರಿಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್, ಪರ್ಟಿಕ್ಯುಲರ್ restriction endonuclease cuts the DNA into number of fragments. ಓಕೆ, ಈ ಫ್ರಾಗ್ಮೆಂಟ್ಸನ್ನು ಮೊದಲು ನಾವು ಸಪ್ರೇಟ್ ಮಾಡಬೇಕು. ಈ ಸಪ್ರೇಷನ್ ಟೆಕ್ನಿಕ್ಕಿಗೆ ನಾವು ಜೆಲ್ electrophoresis ಅಂತ ಕರಿತೀವಿ. Gel electrophoresis, the technique used to separate the DNA fragments. Now what is the criteria here? DNA are negatively charged. That is very important. When this negatively charged DNA loaded on the gel to the gel electrophoresis, they move towards anode under the electric field through medium or matrix. The matrix that we taken here is agarose gel, that is obtained from red algae like Gracilaria, Gelidium, that you come across in the first year chapter. So the first option is, the smaller the fragment size, the farther it moves. ಫ್ರಾಗ್ಮೆಂಟ್ ಚಿಕ್ಕದಾಗಿದ್ದರೆ ಬೇಗ ಆ್ಯನೋಡ್ ಕಡೆಗೆ ಮೂವ್ ಆಗುತ್ತೆ. ದಟ್ ಈಸ್ ಕರೆಕ್ಟ್, ರೈಟ್. ಓಕೆ. ಪಾಸಿಟಿವ್ಲಿ ಚಾರ್ಜ್ಡ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಮೂವ್ ಟು ದಿ ಫಾರ್ದರ್ ಎಂಡ್. ದಟ್ ಈಸ್ ರಾಂಗ್. Then negatively charged fragments do not move. That is wrong. So negatively charged fragments move towards anode. The larger the fragment size, the farther it moves. That is also wrong. So here the criteria is smaller the fragment size, farther it moves. So this is the option, okay, for this question. Then we move on to next question. Okay, so this is the gel electrophoresis. These are the wells, and it contains a matrix, and entire this setup is connected to the electrodes. We have anode here, we have cathode here. When this wells are loaded with DNA fragments, the DNA fragments move according to their size. Smaller the fragment size, faster they move. Okay, larger fragments lag behind, ಬಹಳ ಹಿಂದುಳಿತವೆ. ಓಕೆ, ಇನ್ ದಿಸ್ ವೇ ವಿ ಕ್ಯಾನ್ ಸಪರೇಟ್ ದಿ fragments of DNA by a technique called as gel electrophoresis. Okay. Next question. In the context of observing DNA separated by agarose gel electrophoresis, which of the given statement is correct? ಇಲ್ಲಿ ಕರೆಕ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಯಾವುದಿದೆ ಅಂತ ನೋಡಬೇಕು. ಓಕೆ, in the context of observing DNA separated by agarose gel electrophoresis. See, first option, DNA can be seen in visible light. So this is wrong. You know that DNA cannot be seen by visible light. So this is wrong. Then DNA can be seen without staining visible light. This is also wrong. So DNA can be observed only by staining some markers. Okay, some marker stains. Then ethidium bromide stained DNA can be seen in visible light. Of course, here it is this question partially right, but it is not in the visible light that we can see. Okay, ethidium bromide is the marker or the color that is used to locate where DNA is going in the gel electrophoresis. So ethidium bromide stained DNA can be seen under the exposure of UV light. This is the correct answer. So under the exposure of UV light, the DNA fragments can be seen as bright orange colored bands on the gel that can be isolated by a technique known as illusion. Okay, so answer is D. See, figuratively that has been shown here. Then multiple copies of alien DNA. Alien DNA means foreign DNA. Okay, in the host is possible by the help of one, restriction enzymes. You know that restriction enzymes are used to cut the DNA fragments. DNA ligase is the enzyme used to join the DNA segments with cleaved plasmid to form rDNA. DNA polymerase and DNA helicases. You know, DNA helicase is the enzyme that is used to separate the DNA fragments when DNA undergo replication. But the question here is the multiple copies of the DNA in the host is made possible by DNA polymerase enzyme. Okay, DNA polymerase enzyme, that is the correct answer. Okay, DNA polymerase enzyme, you know, polymerase enzyme adds free nucleotides against the template. That is called polymerization by forming phosphodiester bonds. Okay. Next question. Diagrammatic representation of plasmid PBr322. Full form of PBr, you know, plasmid of Bolivar and Rodriguez, strain number 322. That has been labeled as ABCD. What these ABCD are? Okay, this is the plasmid that you have to remember, at least some parts. See here, these are two selectable markers. Selectable markers. Now, you know that ideal plasmid that is used in rDNA technology should have three sites. One is ORI site, origin of replication, and this origin of replication site is tagged with copy number also. How many copies should be there in the host, that is decided by the copy number. Okay, ORI tagged with copy number. The second important site that should be there in the plasmid are antibiotic resistant site. In case of PBR322, we have two antibiotic resistant site. Here, this is the site, we call it as tetracycline resistant site. So that means the gene that is there in the C region provides resistance against tetracycline antibiotic. Here, this is the region, it is the ampicillin resistant site. This is tetracycline, this is ampicillin resistant site. These antibiotic resistant sites are used as selectable markers. Selectable markers for what? To eliminate non-transformants and selection of only transformants. So that is achieved by using these sites. Selectable markers in the form of antibiotic resistance site. Okay. So here we have two antibiotic resistant site. One is ampicillin resistant site, another one is tetracycline resistant site. Besides this, it also has number of cloning sites. For example, in the region of tetracycline itself, we have certain sites. D is marked. Okay, what is this? What this D? We have to look. Then in this region, there are two recognition sequences, cloning sites. Here PST, Providential Stuarti, Providential Stuarti 1. That is the restriction enzyme that cuts here. PVU. Okay. Similarly, we have PVU second. So these are all the various sites. We call it as cloning sites. The cloning sites may be present on the antibiotic resistance itself or outside that. Okay. Here, what is A, what is B, what is C, and what is D? So the answer is the D, Escherichia coli restriction 1, Hind third, Bomb H1, Sol second. We go into that previous slide. C. So this is this Escherichia coli restriction 1 recognition site. D is the Bomb H1 recognition sequences. Okay. B is the Hind third recognition sequences. Okay. D is the Sol second. C is the Bomb H1. Therefore, the option is D. Option D. Okay. That's correct. A, Escherichia coli recognition 1 recognition sites. B is the Hind third recognition sequence that is there here. And C is the Bomb H1 Bacillus amyloliquefaciens H1 and Sol. Okay, Streptomyces albus. Streptomyces albus. That is the restriction endonuclease obtained from Streptomyces albus. Okay. Then we move on to next question. In insertional activation, the presence of a chromogenic substrate gives dash colonies if the plasmid in the bacteria does not have an insert. Okay. Then presence of insert results into insertional inactivation of the dash and the colonies do not produce dash. These are identified as recombinant colonies. So of course the question is very lengthy one, but answers are simple. There are two ways we can identify the transformants and select. One is by using antibiotic resistance site. Second one is insertional inactivation. Okay, in insertional inactivation, our insert, the desired gene, is incorporated into the coding sequence of beta-galactosidase gene, beta-galactosidase enzyme. So if our insert is incorporated into that region that do not produce beta-galactosidase enzyme and that will not split the lactose into glucose and galactose, that do not produce any color. On the other hand, if our insert is, our foreign gene is not there, then beta-galactosidase coding sequence codes for beta-galactosidase and then they produce color change. That means blue color colonies will be appeared. So therefore, see here, in the insertional inactivation, the presence of chromogenic substrate gives blue colonies if the plasmid in the bacteria does not have an insert. ಅಂದರೆ ನಮ್ಮ ಜೀನು ಅಲ್ಲಿ ಸೇರಿಲ್ಲ ಅಂದರೆ ಅಲ್ಲಿ ಯಾವುದು ಜೀನು ಒರಿಜಿನಲ್ ಇರುತ್ತೆ, ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಎನ್ಸೈಮ್ ಅನ್ನು ಕೋಡಿಂಗ್ ಮಾಡುವ ಜೀನ್ ಇರುತ್ತೆ. ಓಕೆ, ಜೀನ್ ಇಂಟ್ಯಾಕ್ಟ್ ಆಗಿರುತ್ತೆ. ಅದು ಇದ್ದಾಗ ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಎಂಜೈಮ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಇಟ್ ಆ್ಯಕ್ಟ್ಸ್ ಆನ್ ಲ್ಯಾಕ್ಟೋಸ್ ಆ್ಯಂಡ್ ದಟ್ ವಿಲ್ ಟರ್ನ್ ಇಂಟು ಕಲರ್, ದಟ್ ಈಸ್ ಬ್ಲೂ ಕಲರ್. ಓಕೆ, ಸೊ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಇನ್ಸರ್ಟ್, ನಮ್ಮ ಡಿಸೈರ್ಡ್ ಜೀನ್ ನಾವು ಹಾಕಿದಂಥ ಇನ್ಸರ್ಟ್ ಜೀನು ಅಲ್ಲಿ ಇದ್ದರೆ ಏನಾಗುತ್ತೆ? ಇನ್ಸರ್ಷನ್, ಇನ್ಆಕ್ಟಿವೇಶನ್. ಯಾವುದು ಇನ್ಆಕ್ಟಿವೇಶನ್ ಆಗುತ್ತೆ? ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಜೀನ್ ಬಿಕಮ್ಸ್ ಇನ್ಆಕ್ಟಿವೇಟೆಡ್. ಸೊ ನೋ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಎಂಜೈಮ್. ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಎಂಜೈಮ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದಿಲ್ಲ ಆಫ್ ದಿ ಬೀಟಾ galactosidase gene. Okay, the gene that produces beta galactosidase enzyme. Okay, and the colonists do not produce here when the our insert, that means the foreign gene is not there and the beta galactosidase gene is intact, then no color change, that means any color change is obtained. Therefore the option is A. So blue color, beta galactosidase gene, no color, white. This is also called blue-white selection. If the medium shows blue color, then it indicates our gene is not there. Very simple. If the media does not turn into any color, it is intact, white in color, or it is has no change in color, it means our insert is there. So that is the basis, we call it as insertional inactivation. Okay. Next, find the mismatch pair. Find the mismatched pair. You know, Agrobacterium tumefaciens is the bacteria that is present in the soil. It is used to deliver desired gene into the plants. ಪ್ಲಾಂಟಿಗೆ ಬೇಕಾದಂಥ ಜೀನನ್ನು ಪ್ಲಾಂಟಿಗೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಬೇಕಿದ್ರೆ ಇಲ್ಲಿರುವಂತಹ ವೆಕ್ಟರ್, ದಟ್ ಈಸ್ ದಿ ಪ್ಲಾಸ್ಮಿಡ್ ದಟ್ ಈಸ್ ದೇರ್ ಇನ್ ದಿ ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಟ್ಯೂಮಿಫೇಷಿಯನ್ಸ್ ಈಸ್ ಯೂಸ್ಡ್ ಟು ಡೆಲಿವರ್ ದಿ ಡಿಸೈರ್ಡ್ ಜೀನ್ಸ್ ಇನ್ ಟು ದಿ ಪ್ಲಾಂಟ್ಸ್. ಈ ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಟ್ಯೂಮಿಫೇಷಿಯನ್ಸಲ್ಲಿ ಪ್ಲಾಸ್ಮಿಡಲ್ಲಿ ಒಂದು ಜೀನ್ ಇರುತ್ತೆ. ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ TI gene, ಟ್ಯೂಮರ್ ಇಂಡ್ಯೂಸಿಂಗ್ ಜೀನ್. ಸೊ ವೆನ್ ದಿಸ್ ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಟ್ಯೂಮಿಫೇಷಿಯನ್ಸ್ ಇನ್ಫೆಕ್ಟ್ಸ್ ಪ್ಲಾಂಟ್ಸ್, this TI gene normally proliferates the cortical cells to produce a gall that we call it as plant cancer. Plant cancer ಅಂತ ಕಾಮನ್ ಆಗಿ ಕರಿತೀವಿ. ಗಾಲ್ ಅನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ TI gene. ನಾವು ಏನು ಮಾಡ್ತೀವಿ ಅಂದರೆ ಆ TI gene ಅನ್ನು ರಿಮೂವ್ ಮಾಡ್ತೀವಿ. ಆ TI gene ಬದಲಾಗಿ ನಮಗೆ ಬೇಕಾದಂಥ ಡಿಸೈರ್ಡ್ ಜೀನನ್ನು ಆ ಪ್ಲಾಸ್ಮಿಡ್ಗೆ ಸೇರಿಸಿ ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಟ್ಯೂಮಿಫೇಷಿಯನ್ಸ್ ಆರ್ ಡೈರೆಕ್ಟೆಡ್ ಟು ಎಂಟರ್ ಇನ್ ಟು ದಿ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, ನಾ desired genes are delivered into the plant cells. So vector for cloning genes in plants, that is exactly right. Agrobacterium tumefaciens are used to deliver the desired genes into the plant cells. Therefore Agrobacterium tumefaciens act as a vector. Correct. Retrovirus. Okay, retrovirus, you know, these are the viruses that are responsible for causing cancer in animals, that are used to deliver. Disarmed retroviruses are used to deliver desired gene into the animal cells. Okay, but here vector for cloning genes in bacteria is there, it is not correct. So this is mismatched pair. Okay, then biolistic, also called gene gun, it is a delivery of genes without using vectors. Directly microparticles of gold or tungsten coated with the desired gene in small quantities, bombarded with high velocity to enter into the plant cell. That is a vectorless gene transfer in case of plants. So it is through vector to animals. So biolistic, delivery of gene into plants by using gold or tungsten, it is correct. Okay, then micro injection, delivery of gene into nucleus of animal cells, it is also vectorless mediated gene transfer that is used to deliver the desired gene into the nucleus of animal cells. This is also correct. So mismatched pair is here. Okay, option B, retrovirus, their cancer causing genes are removed, disarmed, then desired gene is incorporated and that retrovirus is delivered into the animal cells. So through which we can create transgenic animals. Okay, then we move on to the next question. So this is the retrovirus through which a desired gene can be delivered into the nucleus of animal cell. Okay, but before delivering the gene, the retrovirus should be disarmed. Disarmed retrovirus should be used. Okay. Next question. Before going to next questions, see, there are different methods. I just told, once again recall that one. Method of gene transfer in genetic engineering. One is heat shock method. This is preferably suitable to deliver the desired gene into the bacterial cell. Heat shock method. Second, by using Agrobacterium tumefaciens. Third one is retroviral transfer. Fourth one is microinjection, biolistic, electrophoration, liposuction. These are the two methods of gene transfer that was not mentioned in the NCERT book. Okay. Heat shock method is preferred for bacteria. This is for plants. This is for animals. And these two methods, one is for animals, another one is for plants, without using vector, direct delivery of genes. Okay. See, next question. Find the process of recombinant DNA technology in a specific sequence. So we have some stages of constructing the rDNA, and this were haphazardly arranged. Now adanna ಒಂದು ಜಂಬಲ್ ಆಗಿದೆ, ಅದನ್ನು ಒಂದು ಆರ್ಡರ್ ಆಗಿ ನಾವು ಮಾಡೋಣ. ಓಕೆ. Now first one is the culturing the host cells in a medium. Okay. Then transferring the rDNA into the host, isolation of the desired DNA fragment, then ligation of the DNA fragment into the vector, fragmentation of DNA by restriction endonuclease, then isolation of DNA. So in the first step, the sequence includes, this is the first step. Okay, this is the first step, isolation of DNA. Then after that, you know, fragmentation of DNA, it is the next step. DNA fragmented to make many fragments by using restriction endonuclease. Then isolation of the desired DNA from the fragment. ನಮಗೆ ಬೇಕಾದಂತಹ ಜೀನನ್ನು, ಡಿಸೈರ್ಡ್ ಜೀನನ್ನು ಆ ಫ್ರಾಗ್ಮೆಂಟ್ಸಿಂದ ನಾವು ಯೂಸ್ ಮಾಡ್ಕೊಳ್ತೀವಿ, ತಗೊಳ್ತೀವಿ. Then we move on to transferring the isolation of desired DNA fragment. ಇಲ್ಲಿ creation of rDNA ಇಲ್ಲ. Then ligation of DNA, this is correct. This ligation of DNA fragment into vector, and then transferring the rDNA into host cell, and finally culturing the host cells in a medium. So these are the sequential steps. Therefore, this is the correct answer, option A. So first one is isolation of DNA, then fragmentation of DNA, then isolation of desired DNA fragment, then ligation of DNA fragment into vector, transferring the rDNA into host, and culturing the host cells in a medium. So these are the sequential steps. Okay. So all these steps are mentioned here. Okay. Isolation of DNA, okay, cutting the DNA with restriction endonuclease, mixing the rDNA with that means the joining, finally DNA ligase to seal with the cleaved vector, transformation, entry of rDNA into the host, and cloning finally. Okay, next question. All of the following are correct with reference to isolation of the genetic material from the organisms except, see, one, ಇಲ್ಲಿ ಒಂದು ಮಾತ್ರ ಅದು ಸರಿ ಆಗ್ತಾ ಇಲ್ಲ, ಮತ್ತೆ ಉಳಿದಿದ್ದೆಲ್ಲವುದು that is related to that question. Purified DNA precipitates out after the addition of chilled ethanol. You know, when you remove all the macromolecules from the cell by removing the cell wall and other things, finally DNA is precipitated out in the pure form in chilled ethanol. That is correct. Okay. Next, to isolate DNA from bacteria by treating the cells with enzymes chitinase. So that is treating the cells with enzymes. So bacteria does not have chitinase. Chitin is present in fungus. So it is wrong. Okay, this is wrong. Then to isolate DNA from plant by treating the cells with cellulase, that is correct. To isolate DNA from animal by treating the cells with enzymes DNase. So this is also wrong. Here these two are the wrong, that means wrong statements. A, B and D are wrong here. B and D are wrong. So A and C are right. So except B and D, they are the all A and C are correct. Okay. Then we move on to next question. Select sequential steps involved in PCR. You know, PCR is polymerase chain reaction. It is an in vitro amplification of the DNA segments to make large copies if the amount of available DNA material is very small. Now, what are the steps, sequential steps? Denaturation, extension and annealing. Denaturation, annealing and extension. Extension, annealing and denaturation. Annealing, extension and denaturation. Now, in a PCR procedure, we use thermocycler machine. Okay, in thermocycler machine, the first DNA is denatured. That means DNA segments are separated by treating with high temperature, about 92 to 95 degrees Celsius. When they are subjected, DNA segment separate agat, okay, by dissolving hydrogen bonds. So that is called denaturation. First step is denaturation. Now, these two strands act as a template, okay, template agi work ಮಾಡುತ್ತೆ. Now, free nucleotide sequences should be added and primers should be added. Primers. So, these primers go and bind with the 3' end of both separated strand. Short oligonucleotide primers bind with the 3' end of both separated strand. This is called annealing. Okay, annealing. Annealing ಆಗಬೇಕಾದ್ರೆ temperature 60 degree ಅಷ್ಟು ಕಡಿಮೆ ಆಗಬೇಕು. So first you subjected to high temperature 92 to 95 degrees Celsius. Denaturation occurs. After denaturation, the temperature is brought down to 60 degrees Celsius. Annealing occurs. During annealing, short oligonucleotide primers joins at the 3' end of both the separated strands. That is called annealing. And after that, you know, DNA polymerase extends these primers towards the 5' end. That is called extension. But very important thing that you have to remember is, you know, DNA polymerase that helps in the extension, polymerization, does not work at high temperature if ordinary DNA polymerase is taken. But there is a specific DNA polymerase that is taken from Thermus aquaticus, a bacteria that grow even at 100 degrees Celsius. Thermus aquaticus, the bacteria that grow at high temperature. That DNA polymerase can withstand high temperature. No damage, no denaturation of DNA polymerase occurs if we use Taq polymerase. Taq polymerase means Thermus aquaticus polymerase. So that helps in the extension. So the sequential steps during PCR are denaturation, annealing and extension. This is the correct answer. Okay, we move on to next question. The PCR, polymerase chain reaction, was popularized by which of the following? Okay, easy availability of DNA template. Option B, availability of synthetic primers. Availability of cheap deoxyribonucleotides. Availability of thermostable DNA polymerase. So this is the correct answer. Thermostable DNA polymerase that is taken from Thermus aquaticus. So they can withstand high temperature, do not undergo denaturation, and the PCR process will be completed. So if we use the DNA polymerase taken from the ordinary organism that grow in normal conditions, the DNA polymerase undergo denaturation. So at high temperature, they undergo denaturation. So therefore, because of this thermostable DNA polymerase that is there in the Thermus aquaticus, PCR technique is possible. PCR means in vitro amplification of the DNA to make many copies if the desired genes are present in small quantities. Okay. Then next question. In PCR, multiple copies of gene of interest, that is the desired gene, is synthesized dash using dash and the enzyme dash. Okay. So here answer is, see, in PCR multiple copies of gene of interest is synthesized in vitro. So in contrast to in vivo. In vivo means naturally. In vitro means in the laboratory, inside the test tube, inside the conical flask. In the laboratory we call in vitro. Laboratory alli ಏನೇ ನಾವು ಪ್ರಿಪರೇಷನ್ ಮಾಡಿದ್ರೆ ಅದಕ್ಕೆ in vitro ಅಂತ ಯೂಸ್ ಮಾಡ್ತೀವಿ. So it is in vitro synthesized using two sets of primers. We require two sets, short oligonucleotide primers. So two primers are required. Two primers and the enzyme, you know, DNA polymerase. So in vitro, it is in vitro, 2 sets of polymers, and DNA polymerase is the answer. So option D. Next question. Which of the following is a true statement about the DNA polymerase that is used in PCR? Okay. So we have option A. It is used to ligate introduced DNA in the recipient cell. So it is wrong. Okay, you know that. It serves as a selectable marker. That is also wrong. Okay, selectable marker that we use is generally antibiotic resistance site. Then it is isolated from virus. That is also wrong. Then it remains active at high temperature. That is a true statement with regard to PCR. It remains active at high temperature. That is the answer. Then next question. What does a bioreactor refers to? You know, bioreactor is a tool. Okay, this bioreactor may be a small one if we have a small samples in the laboratory scale, or a bioreactor may be a big one, large vessel where more than 100 or 1000 liters of the products can be obtained. So bioreactor is thought of a vessel, large vessel, patre, alva, large vessel where raw materials converted into useful products by using enzyme, by using plant or animal or even microorganisms. That is called bioreactor. So here options are fermentation tank, organisms reacting to a stimulus, tank for biochemical reactions, and nuclear reactors for biological studies. So these options are there. So it's a very simple answer, that is a fermentation tank. Bioreactor is a fermentation tank. So in a bioreactor, you know, all the conditions are properly maintained. pH, temperature, easy availability of oxygen throughout the bioreactor are all made by special conditions. So that is the bioreactor. So this is a bioreactor. See here, stirred tank bioreactor. It has acid base control, pH control, steam for sterilization, and foam breaker. Whenever a reaction occurs, biochemical reactions occurs inside the bioreactor, foam will generate. That has to be break. Foam breaker, flat blade impeller, culture broth, sterile air, all these are the parts that are there in the bioreactor. Okay. Next. Antibiotic resistance genes in a vector are often used for the selection of. You know, antibiotic resistance genes are used for to select the transformed cells. So competent cells, transformed cells, non-recombinant cells, and non-competent cells. The option, right option is transformed cells. Okay. Then next question. Selection of recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate is called. Very simple. So that is the definition for insertional inactivation. So we inactivate the beta galactosidase enzyme producing gene. By inactivating that, we can our insert is there. So that is used to select the transformants. So option is C. Blue-white selection. See this figure here. It appears as blue color. Okay, that means our gene is not present. If it appears white, it means DNA insert is there here. DNA insert. It has no DNA insert. See here, LAC-Z gene remains intact. You know, LAC-Z gene is responsible for producing beta galactosidase enzyme. You know, 3 structural genes in lac-operon concept you have studied. LAC-Z, LAC-A, LAC-I. So LAC-Y. LAC-Y produces, you know, transacetylase. LAC-A produces permease, and LAC-Z produces beta galactosidase enzyme. So if our insert is our foreign gene, our desired gene is inserted into the LAC-Z gene, Lac Z gene do not produce beta galactosidase enzyme and the colony appears as blue. Okay, because lactose split agate and that makes the colony as bluish in color, blue agi appear agate. On the other hand, ನಮ್ಮ ಜೀನ್ ಅಲ್ಲಿ ಸೇರ್ಕೊಂಡಿದ್ರೆ, ಲ್ಯಾಕ್ ಝೆಡ್ ಜೀನು ಹೊರಟು ಹೋಗಿ ನಮ್ಮ ಜೀನ್ ಸೇರ್ಕೊಂಡಿದ್ರೆ, ಇಟ್ ವಿಲ್ ನಾಟ್ ಪ್ರೊಡ್ಯೂಸ್ ಬ್ಲೂ ಕಾಲೋನೀಸ್ ಬಿಕಾಸ್ ಲ್ಯಾಕ್ಟೋಸ್ ವಿಲ್ ನಾಟ್ ಸ್ಪ್ಲಿಟ್. ಲ್ಯಾಕ್ಟೋಸ್ ಸ್ಪ್ಲಿಟ್ ಆಗೋದಿಲ್ಲ, ಇಟ್ ಅಪಿಯರ್ಸ್ ಆಸ್ ವೈಟ್ ಆಗೇ ಇರುತ್ತೆ. ದಟ್ ಇಂಡಿಕೇಟ್ಸ್ ಇಟ್ ಹ್ಯಾಸ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್. That means if it appears blue, it means that it is non-transformed. In that way we select. Elimination of non-transformants can be made and we can select the transformants. This is called insertional inactivation. Okay, next question. Among the following, which feature in vector complicates the gene cloning? See, one, origin of replication. You know, in a vector, I have told there are three regions that is necessary. One is origin of gene. ಇದು ಬೇಕು. ಇದರ ಜೊತೆಗೆ ಏನಿರುತ್ತೆ origin of replications ಅಲ್ಲಿ copy number ಇರುತ್ತೆ. Higher the copy number, it is the better one. Okay, for example, a bacteria has a copy number 100. That means it has 100 plasmids in a bacterial cell. Okay, origin of replication, first side. Several selectable markers. Okay, so if several selectable markers, you can use any antibiotic. That is also a good one. Single recognition site for same enzyme. ಒಂದು ಎಂಜೈಮಿಗೆ ಸಂಬಂಧಪಟ್ಟಂತೆ ಒಂದು ರಿಕಗ್ನಿಷನ್ ಸೈಟ್, ದಟ್ ಈಸ್ ವೆರಿ ವೆರಿ ಗುಡ್. ದೆನ್ ಮೆನಿ ರಿಕಗ್ನಿಷನ್ ಸೈಟ್ಸ್ ಫಾರ್ ಸೇಮ್ ಎಂಜೈಮ್. ಈಗ ಪ್ರಾಬ್ಲಮ್ ಏನಾಗುತ್ತೆ ನೋಡಿ, ವೆನ್ ಮೆನಿ ರಿಕಗ್ನಿಷನ್ ಸೈಟ್ಸ್ ಆರ್ ದೇರ್ ಫಾರ್ ಎ ಪರ್ಟಿಕ್ಯುಲರ್ ಎಂಜೈಮ್, ನಾವು ಸೆಲೆಕ್ಟ್ ಮಾಡಿದಂಥ ಪರ್ಟಿಕ್ಯುಲರ್ ಎಂಜೈಮ್ಗೆ ಮೆನಿ ರಿಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಇದ್ದರೆ, ದಟ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ ಕಟ್ಸ್ ಮೆನಿ ಪ್ಲೇಸಸ್. ಈಗ ಜಾಯ್ನಿಂಗು ಡಿಫಿಕಲ್ಟ್ ಆಗುತ್ತಲ್ವಾ, ಮೆನಿ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಆಗುತ್ತೆ, ಅದನ್ನು DNA ಲೈಗೇಸ್ ಜೊತೆ ಸೇರಿ ಜಾಯಿನ್ ಮಾಡಬೇಕು. ಈ ಪ್ರೊಸೆಸ್ ಎಲ್ಲ tedious, ಡಿಫಿಕಲ್ಟ್, ಕಂಬರ್ಸಮ್ ಪ್ರೊಸೀಜರ್. ಸೊ ದೇರ್ ಫೋರ್, ವಾಟ್ ಈಸ್ ದಿ ಡಿಸ್ಅಡ್ವಾಂಟೇಜ್ ಇಫ್ ದಿ ವೆಕ್ಟರ್ ಹ್ಯಾವಿಂಗ್ among the options many recognition sites for the same enzyme? That is not a suitable vector. Okay, so for a particular enzyme, it should be preferably single recognition sequence should be there. Okay, so that is the answer. Next question. In a typical gene cloning experiment, rDNA was prepared by introducing a selected gene into the PBR322 plasmid. Okay, PBR322 plasmid ಅನ್ನು ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ನಾನು ಮೊದಲೇ ಅದರಲ್ಲಿ what are the sites that are present I have told. ORI site irutte, two selectable markers in the form of antibiotic resistant site was there. One is tetracycline resistant site, another one is ampicillin resistant site. Then the restriction enzyme used was BOM-H1. You know, when you use BOM-H1, ಎಲ್ಲಿ ಇದು ಕಟ್ ಮಾಡುತ್ತೆ? BOM-H1 ರಿಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಕಟ್ ಮಾಡುತ್ತೆ. BOM-H1 ರಿಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಇರೋದು ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನಲ್ಲಿ ಇರುತ್ತೆ. ಓಕೆ, ಅದು ನೀವು. ದೆನ್ ದಿ ಹೋಸ್ಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಸ್ ಇನ್ಕಾರ್ಪೊರೇಟಿಂಗ್ ದಿ rDNA ವೇರ್ ಕಲ್ಚರ್ಡ್ ಇಂಟು ಟೂ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಮ್. ಎರಡು ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಮಲ್ಲಿ ನಾವು ಈಗ ಕಲ್ಚರ್ ಮಾಡ್ತೀವಿ. ಈಗ ಒನ್ ಹ್ಯಾವಿಂಗ್ ದಿ ಆಂಟಿಬಯೋಟಿಕ್ ಆಂಪಿಸಿಲಿನ್ and the other having the antibiotic tetracycline. Okay, we take two media, one is antibiotic ampicillin, other one is tetracycline. As a result of such a treatment, the transfected bacterial cells, ಇಲ್ಲಿ ವರ್ಡ್ ಅನ್ನು ನೋಡ್ಕೊಳ್ಳಿ ಸರಿಯಾಗಿ. ಸೊ while going in the while you are selecting the options in competitive examination, it is very very important ವಿದ್ಯಾರ್ಥಿಗಳೇ, ನಿಮ್ಮ ಕ್ವಶನ್ಸನ್ನು ಸರಿಯಾಗಿ ಪ್ರತಿ ವರ್ಡ್ ಹಿಡಿದು ಓದಬೇಕಾಗತ್ತೆ. ಗೊತ್ತಾ, ಸ್ಲೈಟೆಸ್ಟ್ ಮಿಸ್ಟೇಕ್ ಮೇ ಬಿ ದೇರ್. ಓಕೆ, ದಟ್ ಯು ಹ್ಯಾವ್ ಟು ಅನಲೈಸ್. ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಟ್ರಾನ್ಸ್ಫೆಕ್ಟೆಡ್ ಸೆಲ್ಸ್ ವುಡ್, ಓಕೆ, ಒನ್, ಸರ್ವೈವ್ ಇನ್ ಬೋತ್ ಕಲ್ಚರ್ ಮೀಡಿಯಾ, ಸರ್ವೈವ್ ಇನ್ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಾ ಬಟ್ ಡೈ ಇನ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೀಡಿಯಾ, ಸೆಕೆಂಡ್ ಆಪ್ಷನ್. ಸರ್ವೈವ್ ಇನ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೀಡಿಯಾ ಬಟ್ ಡೈ ಇನ್ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಾ. ದೆನ್ ಫೋರ್ತ್, ಡೈ ಇನ್ ಬೋತ್ ಕಲ್ಚರ್ ಮೀಡಿಯಾ. ಈಗ ಒಂದನೇದಕ್ಕೆ ಬರೋಣ. ಸರ್ವೈವ್ ಇನ್ ಬೋತ್ ಕಲ್ಚರ್ ಮೀಡಿಯಾ. ಸರ್ವೈವ್ ಇನ್ ಬೋತ್ ಕಲ್ಚರ್ ಮೀಡಿಯಾ ಆಗಬೇಕಿದ್ರೆ ಎರಡೂ ಆ್ಯಂಟಿಬಯಾಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಸೈಟು ಇಂಟ್ಯಾಕ್ಟ್ ಆಗಿ ಇರಬೇಕು. ಓಕೆ, ಎರಡೂ ಆ್ಯಂಟಿಬಯಾಟಿಕ್ ಸೈಟ್ ಆಂಪಿಸಿಲಿನ್ ಮತ್ತು ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಸೈಟ್ ಇದೆಯಲ್ಲ ಅದು ಹಾಗೆ ಇದ್ದಾಗ ಮಾತ್ರ ಅದು ಎರಡರಲ್ಲೂ ಸರ್ವೈವ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನಾವು ಏನು ಮಾಡಿದ್ದೀವಿ, ಬಾಂಬ್ H1 ಸೈಟಲ್ಲಿ ಡಿಸೈರ್ಡ್ ಜೀನನ್ನು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೀವಿ. ಬಾಂಬ್ H1 ಸೈಟ್ ಇರೋದು ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೀಡಿಯಮಲ್ಲಿ. ಇಲ್ಲಿ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ನು ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನು ಹೊರಟೋಗಿರುತ್ತೆ. ಯಾಕೆ ಹೇಳಿ, ನಮ್ಮ ಡಿಸೈರ್ಡ್ ಜೀನನ್ನು ಬಾಂಬ್ H1 ರೀಜನ್ನಲ್ಲಿ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೀವಿ. ಹಾಗಾಗಿ ದೇ ಸರ್ವೈವ್ ಇನ್ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಮ್ ಬಿಕಾಸ್ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಾ ಈಸ್ ಇಂಟ್ಯಾಕ್ಟ್. ಆಂಪಿಸಿಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಇರುತ್ತಲ್ಲ ಅದು ಹಾಗೆ ಉಳ್ಕೊಂಡಿರುತ್ತೆ, ಇಟ್ ಈಸ್ ಇಂಟ್ಯಾಕ್ಟ್. ವೆರಾಸ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಮಾಡುವಂಥ ಗುಣ ಹೊರಟೋಗಿರುತ್ತೆ ಬಿಕಾಸ್ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ ಜೀನು ಬಾಂಬ್ H1ನಲ್ಲಿ ಹೋಗಿ ಸೇರಿಕೊಂಡಿರುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ದಿಸ್ ಈಸ್ ದಿ ಕರೆಕ್ಟ್ ಆಪ್ಷನ್. ಸರ್ವೈವ್ ಇನ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೀಡಿಯಮ್ ಬಟ್ ಡೈ ಇನ್ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಮ್ ರಾಂಗ್, ಬಿಕಾಸ್ ವಿ ಆರ್ ನಾಟ್ ಇಂಟ್ರೊಡ್ಯೂಸಿಂಗ್ ದಿ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಇನ್ ಟು ದಿ ಆಂಪಿಸಿಲಿನ್ ಮೀಡಿಯಮ್. ಇನ್ ದಿ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಅಲ್ಲಿ ಬಾಂಬ್ H1ಗೆ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೀವಿ. ಹಾಗಾಗಿ ಇದು ಆಪ್ಷನ್. ದೆನ್ ಡೈ ಇನ್ ಬೋತ್ ಕಲ್ಚರ್ ಮೀಡಿಯಾ ಇದು ಯಾವಾಗ, ವೆನ್ ದಿ ಪ್ಲಾಸ್ಮಿಡ್ ಡಸ್ ನಾಟ್ ಹ್ಯಾವ್ ಎನಿ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್. ಯಾವುದೇ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನು ಆ ಪ್ಲಾಸ್ಮಿಡಲ್ಲಿ ಇಲ್ಲ ಅಂದರೆ ಎರಡರಲ್ಲೂ ಯಾವುದ್ರದ್ದು ಡೈ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ದಿ see here we have a master plate. So this method of selecting the transformants and eliminating the non-transformant by a conventional method by using antibiotic resistant site is called as Ladderberg replica plated technique. ಅದು ನಿಮಗೆ ಇಲ್ಲ, ಆ ಥರ ಮೆನ್ಷನ್ ಮಾಡಿಲ್ಲ ನೋಡಿ. ಇಲ್ಲಿ ಮಾಸ್ಟರ್ ಪ್ಲೇಟಿಂದ ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಫಾರ್ಮ್ಸ್ ಡಿಸ್ಟಿಂಕ್ಟ್ ಕಾಲೋನೀಸ್. ಇಲ್ಲಿ ನೋಡಿ, ಈ ಕಾಲೋನೀಸ್ ಕೆಲವು ರೆಡ್ ಇದೆ, ಕೆಲವು ವೈಟ್ ಇದೆ, ಕೆಲವು ಗ್ರೀನ್ ಇದೆ. ಇಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡಿದೆ ಸೆಲೆಕ್ಷನ್ ಆಫ್ ರಿಕಾಂಬಿನೆಂಟ್ಸ್. ಫಸ್ಟ್ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ಕೆನಾಟ್ ಗ್ರೋ ಆನ್ ಆಂಪಿಸಿಲಿನ್ ಆರ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೀಡಿಯಮ್, ಎರಡರಲ್ಲೂ ಗ್ರೋ ಆಗೋದಿಲ್ಲ. ಇಫ್ ವೈಟ್ ಕಾಲೋನೀಸ್ ದೇ ಡು ನಾಟ್ ಗ್ರೋ ಬೋತ್. ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ಓನ್ಲಿ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ಕಾಲೋನೀಸ್ ಕ್ಯಾನ್ ಗ್ರೋ ಇನ್ ಆಂಪಿಸಿಲಿನ್ ಆರ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಕಂಟೈನಿಂಗ್ ಮೀಡಿಯಮ್. ಓಕೆ, ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ವಿತ್ ನಾನ್ ರಿಕಾಂಬಿನೆಂಟ್ ಇದು ನಮಗೆ ಉಪಯೋಗ ಇಲ್ಲ. ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ವಿತ್ ರಿಕಾಂಬಿನೆಂಟ್ ವೆಕ್ಟರ್ ದಟ್ ಈಸ್ ಗ್ರೀನ್ ಸೆಲ್ಸ್ ಹಿಯರ್, ಗ್ರೀನ್ ಕಾಲೋನೀಸ್, ದೇ ಆರ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ that is the procedure that we follow. Next question. The rate of migration of DNA fragments through the agarose gel is determined by. See here, e question is a little bit thinking. Somewhat we say an application level question or a little bit complicated, but not a fully complicated. See here. No. The migration of the DNA fragment through the agarose gel is determined by the size of the fragment. Size of the fragment is determined by the number of nucleotides. That is very simple concept. Andre, for example, if a fragment has only 2 nucleotides, it is a very short. If it has 100 nucleotides, it is long. If it has 1000, still long, like that. So, the length of the DNA fragment is based on the number of the nucleotides that are present. Okay, see, ratio of adenine to cytosine, it does not play any important role. Ratio of adenine and cytosine does not play any role. Then presence of hydrogen bonds between base pairs, it's also wrong. Okay, then length of the time the electrophoresis unit is allowed to operate, it is also wrong. So the correct answer for this question is the number of nucleotides in the fragment. How many number of nucleotides present in the DNA fragment determines their movement. Okay, so shorter the length means it contains few nucleotides. The long length means it has many nucleotides. So this is the answer. Next question. How many DNA molecules are formed from a DNA template molecule after 4 PCR cycle? Each PCR cycle has 3 sequential steps. One is denaturation, annealing and extension. After one cycle ಆದಮೇಲೆ there are 2 DNA molecules are produced, because to form one DNA molecule it requires a template, 2 primers, DNA polymerase and the raw materials in the form of free deoxyribonucleotides. So these are all the raw materials. After one cycle from single DNA template, how many? A daughter DNA molecule is formed. There are 2 DNA templates, to start with. Therefore, 2 DNA daughter molecules will be formed. 2 DNA molecules will be formed. Now, these 2 DNA molecules contain 4 strands, 4 DNA polynuclear strands. All of them act as a template. So, after the second cycle, we have, after the first 2, then 4, then 8, then 16, like that. They amplified. Now, here, how many DNA molecules are formed from a DNA template after 4 PCR? So, it is after 1, 2, after 2, 4, after 3, 8, after 4, it is 16. 16 DNA molecules will be formed. That is the answer. Okay. Okay, that is the answer, 16. So, each PCR cycle, see here, after the end of the first cycle, we have 2 DNA. Okay? See here, this is the DNA template, this is the primer. But remember, primer is attached to the 3' end of both the separated strand. So, it is another template. From these 2, we have 2 DNA at the end of one cycle, denaturation, annealing, extension. Then, after the second cycle, we have 4 DNA molecules. After the third, we have, what is the formula? 2 to the power of 1, 2 to the power of 2, 2 to the power of 3, 2 to the power of 4. Very simple. Okay? In that way, we calculate any question asked. After that, we can calculate. Okay. Then move on to next. Which of the following statements about cloning vector is false? See here. ORI is a sequence responsible for controlling the copy number of the linked DNA. I have already told ORI is not only used for the origin of the plasmid, but also it is tagged with how many copies should have in a cytoplasm. So, it is tagged with copy number, that is correct. Selectable marker, selectively permitting the growth of non-transformants. So, this is wrong. We have to use transformants. So, transformants can be selected out, not non-transformants. So, therefore, this is the false statement. Then, to link the alien DNA, vector needs to have a single recognition site for the commonly used restriction enzymes. That I have already told. It is better if you have a single recognition sequence for a particular enzyme. The ligation of the foreign DNA is carried out at a restriction site present in one of the 2 antibiotic resistant sites. That is also correct. Okay. Then we move on to next. Which of these is incorrectly matched? Okay. We have gene gun, biolistic, plasmids, extra chromosomal DNA, DNA polymerases, biological scissors. So, biological scissors, you know, it is restriction endonuclease. They cut the DNA from the desired organisms. Also, they cut the vectors, that we call it as biological scissors, molecular scissors. It is not the DNA polymerase. DNA polymerase helps in the cloning, and bacteriophages are nothing but viruses. That viruses infects bacteria. So, here incorrectly matched pair is DNA polymerase, biological scissors. Then we move on to next question. A biologist must first isolate DNA from an affected person's cell before studying the genetic basis of the inherited human disease, sickle cell anemia. Okay? The next step is to see here. Question nodkoli. First isolate DNA from an affected person's cells before studying the genetic basis of the inherited human disease like sickle cell anemia. What is the next step? Restriction enzymes that break the DNA into small pieces at known points. Gel electrophoresis to separate the DNA from the other cell fragments, ligases that make the DNA stick together, DNA polymerase to make new copies of the DNA. So, these are the 4 options that we have. Here, after the isolation of DNA, you know, very next step is making the DNA into number of fragments. Okay? That is by using restriction endonuclease. So, restriction enzymes that break the DNA to small pieces at known points has to be made. Then we have to identify what gene is responsible for causing sickle cell anemia and their study like that. Okay? So, this is the answer. Next, 32. What is the process of separating and purifying expressed proteins before marketing is called? So, before marketing, you know, any product that is obtained from biotechnology has to be separated, purified, etc. Process of separating and purifying expressed proteins is called as downstreaming process. You know, downstreaming process, very complicated process. It requires lot of understanding about the engineering principles, and that purification and separation together we called as downstreaming process. So that is the correct option. Next, which of the following is not a basis of difference between plasmid and chromosomal DNA? Okay, a little bit higher level of question. Okay, not an ordinary simple question. You have to think for this question. See, not the basis of difference between plasmid and chromosomal DNA. You know, in a bacteria, we have plasmids. These are the plasmids, and this is the chromosomal DNA. Now, here not the basis of difference between plasmid and chromosomal DNA. Now, presence of histones. You know, histone proteins are found only in eukaryotes. Histone proteins are totally lacking in prokaryotes. Prokaryoteದಲ್ಲಿ ಪ್ಲಾಸ್ಮಿಡ್ ಆಗಲಿ ಅಥವಾ ಕ್ರೋಮೋಸೋಮಲ್ ಡಿ ಎನ್ ಎ ಆಗಲಿ ಎಲ್ಲೇ ಆಗಲಿ ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ ಅಂತ ಇರೋದಿಲ್ಲ, ಅಸೋಸಿಯೇಷನ್ ಆಗಿರೋದಿಲ್ಲ. ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ ಜೊತೆಗೆ ಡಿ ಎನ್ ಎ ಅಸೋಸಿಯೇಷನ್ ಆದರೆ ನಾವೇನಂತ ಕರಿತೀವಿ ಅದಕ್ಕೆ? ಆ್ಯಕ್ಚುಲಿ ಕ್ರೋಮೋಸೋಮ್ಸ್. ಓಕೆ, ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ ಪ್ಲಸ್ ಡಿ ಎನ್ ಎ ಟುಗೆದರ್ ಫಾರ್ಮ್ ಕ್ರೋಮೋಸೋಮ್. ಆದರೆ ನಾವಿಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಕ್ರೋಮೋಸೋಮ್ ಅಂತ ಆ್ಯಕ್ಚುಲಿ ಮಿಸ್ನಾಮರ್ ಆಗಿ ಅದಕ್ಕೆ ವರ್ಡ್ ಕೊಡ್ತೀವಿ, ಅದು ನೇಕೆಡ್ ಡಿ ಎನ್ ಎ. ಓಕೆ, ಸೊ ಹಿಯರ್ ಹಿಸ್ಟೋನ್ಸ್, ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಹಿಸ್ಟೋನ್ಸ್ is not a thing. Then presence of nucleotides. Okay, presence of nucleotides is common whether it is a plasmid DNA or chromosomal DNA or eukaryotic DNA. In all DNA, the constituents, building blocks, fundamental unit in nucleic acids, you know, nucleotides. Okay, it is the nucleotides. Nucleotides ನ್ಯೂಕ್ಲಿಕ್ ಆ್ಯಸಿಡ್ಸ್ ಅಲ್ಲಿ ಇದ್ದೇ ಇರುತ್ತೆ. ಓಕೆ, ಅದು ಪ್ಲಾಸ್ಮಿಡ್ ಆಗಲಿ, ಅದು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಕ್ರೋಮೋಸೋಮ್ ಆಗಲಿ, ಯೂಕ್ಯಾರಿಯೋಟಿಕ್ ಡಿ ಎನ್ ಎ ಆಗಲಿ, ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಆರ್ ದಿ ಕನ್ಸ್ಟಿಟ್ಯೂಯೆಂಟ್ಸ್. ಸೊ ದಟ್ ಈಸ್ ದೇರ್. ದೆನ್ ನೇಚರ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್. ನೇಚರ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್, ವೆದರ್ ಇಟ್ ಈಸ್ DAMP, DGMP, DCMP, DTMP, DMP, same. So in all cases, DAMP, DGMP, DCMP, DTMP are there. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. In DNA, we have 4 types of nucleotides. Okay, in RNA, we have 4 types of nucleotides, that is RAMP, RGMP, RCMP, RUMP. ಇಲ್ಲಿ ನೇಚರ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸು 4 ಇರುತ್ತೆ. ಅದು ಇನ್ ಎ ಪರ್ಟಿಕ್ಯುಲರ್ ಸೀಕ್ವೆನ್ಸ್ ದೇ ಆರ್ ಫೌಂಡ್. DAMP, DGMP, DCMP, DTMP, also called deoxyadenilic acid, deoxyguanilic acid, deoxycytidilic acid, and deoxythymidilic acid. So, nature of nucleotides idu. Next, linear form of genetic material. I told earlier, this both are circular, not linear. Linear type of DNA found only in eukaryotes. Linear type of DNA with a telomere. Okay, telomere, no, the last part of the chromosome, terminal part of the chromosome, end of the chromosome is called telomere. Okay, but here such in case of prokaryotes we have only circular DNA. So therefore B and C is the correct answer. B and D is not the basis of difference, not the basis of difference. Presence of histones difference and linear form, that is different, because histones are absent, linear form of DNA is chromosomes not there in prokaryotes. Next, spooling is known as spooling. You know, spooling means it is the collection of DNA, isolated DNA. First option, collection of isolated DNA that we do whenever we isolate the DNA from the choice organism. Spooling means isolation of the collection of isolated DNA. So this is the answer, correct answer. Then DNA precipitation out of a mixture of a biomolecules can be achieved by treating with. We have 4 options: chilled methanol, chilled ethanol, chilled butanol, chilled chloroform. You know that this is the answer, very simple, direct answer, chilled ethanol, on which DNA filament precipitates. Why it is DNA fragments precipitates, that can be taken out for further enzymatic digestion. So answer is 2, chilled ethanol. Okay, Stirred tank bioreactors have been designed for, stirred tank reactors, the main intention is to, very simple, availability of oxygen throughout the process. Because the entire process is aerobic one, it requires constant supply of oxygen, so oxygen has to be mixed with the contents thoroughly. That's the main intention of the stirred tank bioreactor. It is mainly designed for availability of oxygen throughout the process. Okay. Microbes found to be very useful in genetic engineering are. Now some microbes are very, very useful. They are, option 1 nodi, Vibrio cholerae and Salmonella typhi. Vibrio cholerae is a pathogenic organism that causes cholera. Salmonella typhi, you know that it causes the typhoid. It's not used. Diplococcus species and Pseudomonas putida. Diplococcus, causes the pneumonia. Streptococcus related species. Pseudomonas putida, of course, used to transform into a super bug that is used to clear the oil spills in ocean, of course. Pseudomonas putida is used, but the most common, very useful tool, very useful microorganisms used in genetic engineering are Escherichia coli, E. coli and Agrobacterium tumefaciens. Most commonly used is Escherichia coli, but Agrobacterium tumefaciens used to introduce desired gene into the plant cells because it has a vector. That vector ಅಲ್ಲ ಟಿ ಐ ಪ್ಲಾಸ್ಮಿಡ್ ಅನ್ನು ರಿಮೂವ್ ಮಾಡಿ, ಟಿ ಐ ಜೀನನ್ನು ರಿಮೂವ್ ಮಾಡಿ ನಮಗೆ ಬೇಕಾದಂಥ ಜೀನನ್ನು ಸೇರಿಸಿ ಪ್ಲಾಂಟನ್ನು ಮಾಡಿಫೈ ಮಾಡ್ಬೋದು. ಸೊ ಬೈ ಕ್ರಿಯೇಟಿಂಗ್ ದಟ್ ವಿ ಕ್ಯಾನ್ ಹ್ಯಾವ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, ಸೊ Escherichia coli and Agrobacterium tumefaciens are the most commonly used. Even Saccharomyces to some extent used, but these are the common. Okay, when we have two options, we have to select most appropriate answer. Okay, ಎರಡು ಆಪ್ಷನ್ನು ಆಲ್ಮೋಸ್ಟ್ ಕರೆಕ್ಟ್ ಇದ್ದಾಗ ನಾವು ಅಪ್ರೋಪ್ರಿಯೇಟ್ ಆನ್ಸರನ್ನು ಸೆಲೆಕ್ಟ್ ಮಾಡಬೇಕು. ಓಕೆ 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 I will end with this session with the biotechnology processes and principles. Students, please go through today itself, read this biotechnology and the first chapter, principles and processes. At least some concept you will recall it. I know that you are in exam mood state, still in the exam mood state, and the time availability is very short for your CET and NEET. Please prepare carefully, please prepare thoroughly, and you will become success. I will take another chapter today itself, that is applications of biotechnology. Okay, thank you.